I've got plenty to share, including my microscope here, which I hope I can do a quick demo on. So I'm going to share my screen, and I've got this PowerPoint thingy here. And I had to start with this one, of course. So this is Whitney, Robert, and Sarah, all staff at NOFA, New York, take in a beautiful summer day, just so we can remember what that was like, not that long ago. And they're all learning microscopy here. So at that happy note, let me just go and start the slideshow. All right, so I'm basically going to be talking about soil and compost under the microscope. We're going to take a look at that. We're going to talk about the soil food web. I studied under Dr. Elaine Ingham and learned so much, and would suggest looking at that program. They have a number of courses available if you really want to get into it. So we're going to take a look at life in the soil, what it looks like, and also how you can get it, what things you can do in the soil, mostly, of course, regenerative ag principles that will bring more life into your soil. Okay, so I'm going to move on. This is a true mentor of mine. This is Vandana Shiva, and of course, her wonderful comment. And if you don't know of her, she's doing amazing work around the world on seed sovereignty, and she's a brilliant woman. Her comment, in nature's economy, the currency is not money, it is life. And of course, I would have to agree. Okay, so we're going to go to the next one. How do I do that? There we go. So I always start with, I'm going to bring this a little closer. I don't have to reach so far. Okay, we're still good. I like to start with the scary stuff about the nutrition in our food and how it really is not good, and compared to years ago, it really has dropped. And it depends on how the food is grown. We're finding a number of articles. I know you all know this, the decline of the nutrition in the food in the last 50 years, and that really is attributable to the chemicals that we are applying. And so, for example, if you look at the difference between organic and conventional, it's a huge decline in conventional. Just because the microbes, the life in the soil, is killed. And what does that possibly have to do with that? Well, I'm going to talk about that. And this is really the premise of why it's important to have life in the soil in the form of microbes. Soil health is basically, I've got to move us here so I can see. Hold on a second. Read this. The continued capacity of the soil to function as a vital living ecosystem that sustains all of us. So this is NRDC showing us the difference between dead and living soil. Basically, if you see bare soil, chances are it's dead because there's nothing to protect it. And of course, it'll erode, and you'll need increased chemicals to grow anything, and that continues to deplete the life in the soil. So comparing that to living soil, as soon as you see earthworms in the soil, you know, being the highest trophic level in a soil food web, that you have life in that soil. Oh, it's going on by itself, isn't it? Uh-oh. Okay, so yeah, this is a Bionutrient Food Association. They have some really fascinating talks on their website if you want to look them up. And basically they talk about nature and where plants produce sugar through photosynthesis, and they feed take it in, and up to 50–70% sometimes of that, what we call exudates, gets fed to the beneficial microbes that are in the soil. And here's a really nice look at what that looks like. So the plant is feeding the microbes, and in exchange, the microbes will get whatever nutrients that plant has the potential to take up and feed it back to the plant. So that is why that symbiotic relationship is what makes fertile soil. So this is a wonderful picture from Dr. Ingham, where she talks about, she likes to call them cakes and cookies because they are sugars, and the symbiotic relationship that, oh, I'm just having to admit people here, hold on, without moving forward with the screen. Let me go back. Okay, hold on. Okay. So it jumped to this next screen. I think it wants me to hurry up. This is something that you might have seen before, the soil food web. And we're really talking about the different elements and trophic levels within that soil. So we're going to take a look at what that looks like, the fungi, the bacteria, and then the predators of those, the protozoa, the nematodes, the microarthropods, and we're really going to see what they look like in the soil. The other thing we're going to check is the balance between fungi and bacteria. So generally, as the minute you plow the soil or you add chemicals or you disturb it in any way, you are going to have bare soil, or you have bare soil. It's going to be 100% bacterial. And so you're selecting for weeds. If you go into the old growth forest and you take the soil there, you're going to find it's really rich in fungal, and you're selecting for trees or perennials. So what we want is to find a balance of bacteria and fungi so we can grow what we would like to grow, which is row crops, grasses, pasture, vegetables. So that's why it's important to have fungi in our microbial population. So of course, different plants need different soil microbes. So if you're talking about brassicas, they can handle more bacteria, while as you go closer to the woodland edge, things like strawberries and other fruits and perennials like it more fungal. So let's take a look at what that looks like. I'm always doing trials of no matter what I'm doing, I have to trial. I have a control, then I have one where I'm adding compost, I'm adding compost tea, I'm doing various studies of what the life looks like. So here I took, in this example, 2 pots, put the same seeds in there, and one is in just soil that I bought at you know where, with chemical fertilizer, we'll call it, and one is just organic soil with some worm castings. So here we are, I think this was 2 weeks later, and then another 2 weeks. Look at the difference in the color. Look at the harvest. So the one on the left, that what I did manage to harvest in terms of lettuce was sort of chewy and not a lot of taste. The one on the right, sweet, crunchy lettuce. So already I was starting to see effects. And then here we are 2 weeks after that, and the disease had gotten into the one with chemical fertilizer. So the reason I'm showing this is the effect above ground. What does it look like below ground? So under the microscope, this is the bag soil with the chemical fertilizer. And you can see there's no soil structure. There's a little bit of organic matter in there, but generally it's pretty dead. That's a ciliate, which is not what you want to see. So that is an anaerobic microbe. And the bacteria are not forming aggregates as they should. So if you compare that, this is the bag soil. There's a nematode in there, and you can see how rich it is. And so bacteria, when they're acting normally, are sticky, and they will form these glues, and that forms these aggregates, and that gives you soil structure. So when you do put any water through there, it holds that, and it also holds nutrients. So this is what you want to see in terms of living soil. So let's take a closer look at what that looks like. Now, microbiologists are saying there's like 1.5 million species of soil fungi and 3 million species of soil bacteria in healthy soil. But what we're finding with the agricultural soil, those species and numbers are greatly declined. And that's that soil food web again. And now we're really going to take a look at what that looks like under the microscope. So you can see bacteria are small. They're either round or rod-shaped, cocci, or rods. And then the fungi, and there's a beautiful strand of fungal hyphy right there.
Monique, we have a quick question, if
Yes.
I can interrupt. Don Carlson asks, is there a correct percentage of worm compost to soil vegetables and herbs?
With worm castings, what I tend to suggest when you're seed starting, I put in 10% worm castings. And then when I'm transplanting, I'll put maybe a handful for every transplant. And so what that does is the microbes that are so plentiful in things like really high quality compost, which we're going to look at, or worm castings, go around those roots, and it starts that symbiotic relationship right away, right when that transplant is going in the ground. And then as those roots develop and grow, the microbes multiply and follow the roots. So that's the key time to apply it. I wouldn't suggest taking worm castings and throwing them on your field and saying, well, good luck, because again, bare soil, but putting them around roots where that symbiotic relationship can start right away is the best use of high quality compost. Or, well, compost is different, which I'll talk about later, but certainly worm castings. And then making a compost tea from the worm castings is what I would recommend. So I hope that answered that question. If not, ask me another. So here's another beautiful strand of fungal hyphae. And right along. Come on, there you go. We talked about that. Is it going backwards? No, okay. I guess I duplicated those slides. This is the smallest predator. It's a type of amoeba. It's called a flagellate. And you can tell, you'd love to see these. Again, we're zoomed in 100 times, although it looks, this is 400 times, I'm sorry. Oh, who did these slides? Anyway, it tends to bumble around and is collecting and eating that bacteria as it goes.
There's another question in the chat. Fiona asks, is there something added to the soil before you look at it under the microscope, or is it just bare soil?
What we're looking at now? It could be really high quality soil. Often it is, sorry, I'm gonna take a drink. COVID recovery here, doing well though. Often when you find the most microbes, especially the predators, it's in a type of compost or worm castings. But this could easily come from soil. It's just they're less numerous, so you have to go through a lot more. But often with one drop of really high quality compost, you can find a lot of predators, as we're going to see here. So this is another type of amoeba. This is a testate amoeba, the test around it. And this is a nematode. There are many types of nematodes. Generally, we find the bacterial feeding nematode, which is wonderful because that nutrient cycling, the nematode comes in and what it puts out are those plant available nutrients that are in the bacteria. So it releases those. So that's why it's so important for nutrient cycling to see things like nematodes. There's again, some beautiful pieces of fungal hyphae there. All right, we're going to the next one. Yes, we are. Huh, it's funny that it's acting up here. I wonder if I have to stop sharing and share again. Not sure why it's not letting me go to the next. Let's see. Leah raised her hand. Oh my. Are we seeing that?
Is it also flipping?
Yeah, I'm seeing, oh my, there's a lot of action going on here. So yeah, I'm seeing some flipping going on.
Yeah.
Leah, you guys have questions?
Yeah, Leah, if you have a question, feel free to unmute and ask. We see your hand up and down.
Maybe that was an error as well. I wonder if we're having technical issues, but.
Yeah.
All right, I'm going to try sharing again, see how we do. All right, there's that guy. Moving on. Okay, so I'm going to escape here and go to the next one physically. Okay, share my screen again. All right, you seeing the next one? This wild and crazy nematode, we're zoomed in 400 times. You can see all the bacteria that this nematode has consumed. And also take a look at these beautiful aggregates. This is really high quality soil, a lot of bacteria in here. That's what you want to see. And of course, it's always nice to see these types of predators here. I love these guys. These are micro-arthropods. And this guy, quite large, stuck under the slip cover. They look prehistoric to me. And they're a really good sign that you have high level of soil in the various trophic levels in your soil. Here's another guy. So we're going to zoom in on him. Now we're looking at things that you might find in anaerobic conditions. So often I'll see these in things like compost tea that's gone anaerobic. You let it sit and there's not enough oxygen, or soil that's been inundated with water and it's pooling. So you don't necessarily want to see these guys. They will consume a lot of the bacteria and just burn it off, and so that there will be less and less microbes available for plants. So this one is from some compost tea when I was at Dr. Ingham's farm. We were doing a lot of samples, and someone sent in this sample and it's full of ciliates. So they've consumed all the bacteria and basically not what you want to add to your soil. One of the main reasons to do microscopy is to make sure, especially if you're making compost teas or doing extracts, that you don't have this anaerobic condition happening. But we'll take a look at that in a little bit. This I found, again, not a great sample. If you see these, it means you have anaerobic conditions. It's a type of ciliate, but it's so beautiful, right? We're zoomed in. Imagine this going on. We're zoomed in 200 times, and we have this amazing ciliate that should keep going. There we go. Did I include the whole thing? He should let go of it, start rotating around. There he is. He didn't like whatever he ate there. But it really does remind me of the incredible life that is going on in this soil. I mean, we're zoomed in 200 times. He's. I just gotta go to the. Did you have a question? I love that voice. Anyway, we lost our ciliate, but I found him later stuck between this aggregate and organic matter, still revolving around full of bacteria. So I just think that's a beautiful sight. Although it does indicate our anaerobic conditions, as does this guy. This is an insect larvae. So if you have, say I have my worm bin over here, and I just throw in some apple cores and things, I look inside, now there's fruit flies, those fruit flies are gonna lay eggs, and now you're gonna end up with these sorts of things. Not only will the bin be anaerobic, but you'll start seeing these anaerobic type microbes like this insect larvae, who's doing a very good job destroying the aggregates there. Here's another one you can see. This is a rotifer, and it should start. Why is it not playing? Okay. So what you're seeing here, this is a potworm, a baby potworm, and this is a nematode. So you can see the different size, the size between a tiny worm. You wouldn't even see this with your without a microscope. And just comparing that, and also the efficiency of the worms going through this organic matter and breaking it up, so making it more and more aerobic, and also getting that nutrient cycling done. Very efficient. Worms really are our best friends. This next one.
Monique, we have two questions in the chat, both the same, and it was Fiona's clarification of her early question, asking, I was wondering how you prepare the soil before looking at it under a microscope.
Oh, we're going to do that. I have the microscope here.
She said if I wanted to try to look at soil under a microscope as an amateur. So it looks like it seems to ask more of the technical question of the steps, like how. Heather, did we lose?
I do not see Monique. Yeah.
Monique? Okay.
Oh, there she is.
Oh, there we go.
Didn't like it when I turned the computer sideways, did it? Sorry about that. Okay, go ahead with the question.
It was Fiona was asking a clarifying question, asking how, like, it seems like what the steps would be to look at soil under the microscope. Like, is
Right.
it a drop of soil, put water in it, do you shake it around, the steps?
Okay. So if you hold on, how are we doing for time? We're half an hour in. So I'll probably get to that in the next 15, 20 minutes.
Awesome.
I'll do a demo of that, but I'll be very careful moving my computer apparently, although I know where to find you. Okay, was there another question?
It was the other question was the same as that one.
Okay, cool. So we're going to take a look at that. All right, so let's go back to our wonderful, I'm going to go to this one here. It's funny, the technical glitches. Is it the cold? All right, so there's a nematode. Let's see what you can find in this drop. So there's a lot of life in here. We have nematodes, microarthropods. Oh, I think that's a nematode there, right? Can you see anything else moving? Oh, nematode, microarthropod. So lots of activity in this one drop of excellent compost. So let's move on to the next one. Okay, so we've done that. I'm just going to go to the next one. For some reason, the videos might be making it hang up. So play from that. So now you're seeing Mac on the microscope here. I'm assuming you are. Thanks, Alex. So I do have a few photos. One of the things that I love to see are farms that are getting more and more into their soil. And once you take a look at it under the microscope, you can never go back, because then you really know what you're seeing. And it does tell you so much. So again, back to this slide. So we managed to get a grant so that the staff in the different regions of New York, NOFA staff, could now take this microscopy knowledge and go to farms and be able to use that. So there are different ways to bring that soil to life and to add as many microbes as possible. And so I'm going to go through a few steps. So for example, try to avoid any type of chemical or physical or biological stressors. So chemicals, obviously, pesticides, fertilizers for obvious reasons, and manual tilling. Zach Bush, who's a brilliant endocrinologist, says rototilling really is as equally as damaging to the soil as spraying chemicals. So trying no-till whenever you can. And diversity. The more diverse the plants, the more diverse the life is in the soil. And then if you can, I think one of the keys, which I learned from farmer Craig, who runs Coogan Farms, which is a giving farm. All their food is donated. So Craig Coogan, if you can look him up, he talks about always trying to have things living in the soil year round, or as long as you can. So perennial plants, ground covers, cover crops, and of course intercropping, which we'll talk about, and trees and shrubs. So if you can see the soil, it really means that it's losing its carbon, it's losing its life. So what you want to see is the soil covered, especially in the form of cover crops, which is really the best way to regenerate the soil. So this is a lot of words, and I'd rather do the demonstration. This is going to be available if you want to look at all of the benefits of soil biology. You can take a look later on that. And again, this Dr. Ingham, the only way to get things back in balance is to reintroduce that life. And you can do that through properly made compost, which you're going to look at, worm castings, and compost teas. So when you're talking about compost, that really is an incredible way to bring those microbes that are existing and have done the nutrient cycling in compost and putting it on your soil. So I can't go on enough about the importance of compost and doing your own composting. So you have indigenous microbes from your soil and from your residues going back into the soil. And of course, a diverse plant community. So as Elliot Coleman says, producing quality compost is the most important job on an organic farm, and I would agree. I do a very long lecture on composting and compost tea, but I'm just going to give you a little preview here of the various composting methods that you can do. And the main rule of thumb, if you will, for any compost is knowing the carbon to nitrogen ratio of what you're putting in. So high carbon materials mixed with high nitrogen materials, so that you get what we want is a 25 to 30 parts carbon to nitrogen. So again, this is a long story, so I'm just giving you the highlight. If you have too much carbon, it's just going to probably sit there. So imagine wood chips take forever to break down. If you are putting just food scraps in, it's going to turn into a stinky mess pile, and it will go anaerobic, and because it's too much carbon, nitrogen. So it's that balance that's the key to doing composting correctly. All right, so we're going to take a look at compost under the microscope. I have if I have time for this, I think I do, a 10-minute video showing compost from various producers and what it looks like under the microscope. So let me go to this video and I'm going to start playing it.
We're here at Earth Care Farm. We've been making compost for a little over 40 years. We have a large diversity of ingredients that we start our compost with, from bedding and manure and leaves, wood chips, coffee grounds, a lot of fish scraps and shellfish, and some food scraps as well. And we have a longer process. It takes us about a year to make a finished batch of compost. And then we do screen it to a half-inch screen, half-inch or less. And at the end, we have this black, beautiful, life-filled goodness to spread onto your gardens and fields.
100 tons. This is compost from Earth Care Farms. You can see it's very full, but what I am seeing is not the fungal. Loads of bacteria, very diverse. Aggregates throughout, so nice, mature. No predators that I could find, but active and alive. So it is definitely bacterial dominant. A nice assortment of various types of bacteria.
The scale I'm able to achieve here by having this equipment, I'm able to process hundreds of tons of material a day, turn that into a rich living soil. My impact is so much more. So don't be intimidated by the equipment. That's something you can figure out. It's not hard. And it's really empowering.
Okay, my name is Domingo Medina. I'm the founder and director of operation of Peels and Wheels Composting. It's basically a composting service. We pick up food scraps that the residents and households produce, also schools and small businesses and restaurants around New Haven. We collect them through a system of bikes and trailers, and we bring it here to gather New Haven, to the former New Haven Farms at the back, the parking lot of Phoenix Press, that has allowed us to use this space for farming and also for producing compost under the concept of community composting. I work with what's called aerated static pile system. These are in vessel, and what it means is that we supply oxygen, air, through a system of PVC pipes in the back of the system. We collect energy from the sun with solar panels. We store that energy in batteries, and then we have an inverter that will change the DC voltage to AC that runs a blower that allows to push air into the chambers. These systems are great for urban settings because they are enclosed. Once it's in, we have the proper recipe, we have the proper moisture, and the rest is pushing it into the chambers. In a matter of 30 days, we take this material out and we put new material in.
Oh, crap.
This is sifted compost. So this is the end product that we really like, and something that we're learning, not only the properties of this compost, but the soil biology that we might be producing.
Look at all the fungi
the
in
soil
that
biology
compost.
that we need to be able to guarantee all the beneficial properties of compost. Most importantly, the holding capacity of nutrients and the cycling property of the soil biology in the soil food web of your compost. Those are, I think, the major benefits that compost can do for your soil.
What we do is, because of the fact that we grow the microgreens, and so we have so much of this
So he takes the finished trays
soil
and he
that
recomposts.
we dump every week. So we basically are dumping about 3,000 plats a week. Most of our customers are either landscape gardeners or companies that are building gardens for homeowners, and are using this in mostly raised beds because it is so rich in organics, and it is certified organic. So the homeowners want that now. But you can see this is probably a year old, and it's, look how rich it is. You can see how rich it is and how dark it is. I mean, see the difference between the sun hitting it,
This is 100 times magnification, finished compost from Gilberti's. See those small rods? Those are Fungal Hyphy, and I'm seeing excellent fungal throughout this drop. A lot of organic matter still to be decomposed, and no predators that I can tell. And look at that beautiful piece. Let me just zoom in. That's 100. This is 400 times magnification. Nice aggregate structure there. Sandy, you can see those glass bits. That is sand. So there's nice septa along that fungal hyphae there. Partitions between the cells. And in the background, you can see good diversity of bacteria. You have some rods and some cocci, round cocci.
Well, we operate Ossawaga Farm in Putnam, Connecticut, and we grow a diverse range of organic vegetables. And what else?
We're no-till. This is our 4th season.
Our practice is emphasize the soil health. So we try to really think about the living ecology in the soil, whatever we do.
So we're doing thermophilic composting. We measure out the ingredients with 5-gallon buckets, and we'll do a 30 or a 40 bucket pile. Pretty much every week we've been doing it. So we've got several piles here that are in different stages. One we just made yesterday, this one here. So you can see it's nice and green and fresh. After a few days, it's gonna not be green anymore. And we'll turn these twice, get them up to 130 degrees or more. And that one is still coming up. I think it's just above 120 right now. So it'll probably hit 130 tomorrow. And then once it hits that temperature, we let it sit for 3 days or 2 days or 1 day, depending on how hot it got, just to make sure that we've cooked out any pathogens and weed seeds. And then we'll flip it, make sure that we put another part of it into that hot center. And we'll do that twice so that every part of the pile cooks. And then we'll monitor the temperature, turn it if we need to, but just let it come down. And once it's come down, we've got a pile like this, which is something that we would use in the field. And rather than that deep compost application, this is more of an inoculant. So if you smell this, it smells really sweet. There's a lot of biological activity. If you look at it under a microscope, which we do, you can see the bacteria, the protozoa, the fungi, the nematodes, all the good guys.
100 times magnification. This is from Ossawaga Farm, that compost pile that's just finished. You see it is really teeming with fungi, various types of fungi, which is really good to see. Some like fungal spores in the center there, these circles. Let's zoom in on this piece here. So now we're looking at 400 times magnification, you can see how diverse all of this fungi attached to this aggregate here. So this would make excellent amendment to any soil, especially if you were planting more perennials that need a higher fungal ratio to bacteria.
Okay, I'm going to stop there because I know there is some interest in doing the microscopy. So I do want to show you that. I'm a little nervous moving my computer because I don't want to lose you again, but if I do, I'll be right back. Okay, so I'm going to turn this very carefully over here where I have my microscope. Okay, so this is a microscope. It costs about $324 approximately. It's the one I recommend. And the reason I like this is that I could show you all these videos of what it looks like. There's a connector here that goes, takes the video or photos and you put it into, I've got a MacBook Pro thing. So then you just plug it in and go to photo booth and it shows up there. So I'm going to do that. But first I'll show you how to prepare a slide. So you take a test tube. I have some compost here. So I'm just going to fill up what we call 1 gram. So I take from, if I was doing this around soil, I would go around the roots of the soil. So remember, this is compost and it's not necessarily interacting with soil and it's been sitting around. So I'm not sure what we'll get, but let's just check it out. I'm more interested in showing you how to make a slide. So you've got where it changes shape, that's 1 gram. And then I have distilled water over here and I fill my pipette and I'm going to do a 1:5 dilution, which is basically 1 gram to 4 grams of water. And that's what they call 1:5. I call that 1:4, but I'm not. All right, so there is our solution. And I put the lid on, and then I do the same process regardless of what it is, any type of soil or compost. And so that you know it's been mixed the same amount of time. So this is how I do this. This is going to get really boring, but I'm going to count to 30. And I want to, every time I do it, release the microbes from, I hope someone's counting. Alex, can you count while I'm talking? So what I'm doing is and having it go into the liquid without smashing it too hard, but definitely feeling like it's being released. If I'm being too wimpy about it, they won't come loose from the aggregates in the compost. How are we doing, Alex? You're muted.
Now we're at 30.
Oh, perfect timing.
Yep.
All right, so I'm going to stop. So there's your solution there. I have a slide and a flip cover, and I'm going to take a clean pipette, go down about halfway, let about half of it back into the solution, and put 1 drop on my slide. Now, if this was, and then I have another thing of water, which is my mixing water, because I don't want to mix this up. So that's, just believe me, you want 2 things of water. Because the one will always be where you get the clean water. So then you take your slip cover, put it on top, and you want to see it spread out evenly across the entire. So I don't know if you can see that. And then we're going to put it under the microscope. I'll take this one out that I was looking at earlier. And then I'm trying to remember how to do this. I'm going to be plugging this into my computer, USB port, and then I'm going to open up Photo Booth and share my photo booth, and you should see what it looks like. All right, so go back to you guys, share screen, Photo Booth, share. Okay, turn the light on. Cool. We're at 40 times magnification here. So this is the compost that we're looking at in real time. Isn't that cool? So that's really full. So it's hard to tell at 40 times magnification And this is really quite dense. I did notice as the drop went down, it didn't spread out as evenly as I would have wanted, but this is now 100 times magnification. So really great aggregate structure there, but very difficult to see. It's maybe a little too thick, too many layers on top of each other. Let's see here. Let's see if we can zoom in on that. This is 400 times magnification. There's a piece of fungal hyphy there. A lot of organic matter. So really great aggregate. So you can imagine the nutrient retention and water retention by having all of this life around the roots of your plants. Compared to that earlier image that we saw of the chemical soil, which was basically barren. The structure here and the density of it is really intense. So that's how you prepare a soil slide.
Monique, Glenda Neff asks if you can use your cursor to point out quote unquote organic matter.
Certainly. Okay, so here we are. Organic matter in this image, see this? Anything that is not, like these are aggregates and you can tell that they're made up of other things. But when you see things like this, you can probably see better when I zoom out, that those are all, that's organic matter. You can also see that the activity going on. So I also look for that subtle, that the whole thing seems to be dancing almost, if you will. If I go back to 100 times, it just takes a while to change focus. I'm going to go a little bit on the outside of this drop because it's so thick. Here we are. This is a little less intense here on the edge. All right, so this is 100 times, so everything's of course smaller. That's a fungal hyphy. This would be organic matter, probably organic matter here. Not seeing any predators. Sometimes it does take some time to find them. Here you have fungi. This is probably organic matter. This is just a giant clump. So I could do it again, but I did more want to show you how the simplicity of making a slide. If you were doing compost tea, you would just go into the tea and add one drop and look at it under the microscope. So there's no type of, sorry, I just have to take a drink. All right,
there's a couple questions in the chat. I don't want to miss them.
Okay, go ahead.
Where we were. Jeffrey asks, and this is from a little bit earlier, so my apologies for missing this one. Jeffrey asks or states, I've been hearing about an invasive worm in New York State. Do you know about this creature?
Yes, the Asian jumping worm. I've seen them everywhere, including Block Island, the Berkshires, all around here. Where there's a conference coming up. If you look up Jumping Worm Conference, I think it's January 25th or 26th. They're going to be discussing it. So it's becoming a really well-known concern. And these worms, you can spot them. They tend to be longer and smooth, and they have a very distinct white band. So if you do see that, try to get them out of there. And what I'm going to do in any field I'm working at this spring, if I see them, definitely take them out before they can start mature and start leaving cocoons, because the cocoons can apparently overwinter. So the one good thing about this cold winter, it might kill the jumping worm cocoons that we're all dealing with, because it's so freaking cold. So that is the hope anyway. So there is more information of the Asian jumping worm if you want to look that up.
Alrighty. And next question from Tony asks, is organic matter that composts in soil sufficient to add life to the garden?
That's an amoeba, by the way, with the test. What was the question?
Tony asks, is organic matter that composts in soil sufficient to add life to the garden?
Yeah, in organic matter, there will be life, because guess who's eating that organic matter? The microbes and worms and other smaller creatures. So, you know, there was always a philosophy. I remember early on with more conventional, people thought there was a limit to how much organic matter you could add. I don't think there is anymore. I think we're finding the more organic matter, the better for your soil, and to give food for the microbes, and to add obviously soil structure, and as it breaks down, it's going to be adding more and more nutrients into the soil.
Sue and Carrie both ask, what brand of microscope do you have? Like the make model.
Okay, how do I get this to you without losing my train of thought? It's an Omax, because I have a link to it. If you type in, where did I get it? On Amazon, the Omax microscope, compound microscope with video connector, and it's $324. I know that, but I can't look it up right now. So hopefully you can find it.
Then Megan asks, when you're referring to aggregates, what is the aggregate an aggregate of?
Oh, okay. So you see these guys here? See this? There's a flagellate, by the way. And these, the aggregates are made up of bacteria. Bacteria are sticky. So if I did the whole talk on compost tea, I would tell you about spraying the foliage, because the bacteria will stick to the leaves and will eat any, for example, if you have powdery mildew or something, it'll eat the powdery mildew and protect the leaves. So in this case, they are sitting in this compost, which is mature. They've gotten together and formed these aggregates. When I look at compost that is less mature, so for example, Alex and Yoko's soil compost that they had just finished, you didn't get this complex soil structure or aggregates because it hadn't had a chance to form them. So over time, they add that structure. When you put it into your soil, you'll have these aggregates that are really just bacteria.
And last one in the chat that I see for right now, Shannon asks, I'm going to butcher these scientific names. Adaintium jumping worm, same as Asian wiggles, and Asian jumping worm, same as Asian red wigglers. Like asking if those two are same.
I've never heard of Asian in front of red wigglers. I don't know if they are in fact Asian, but the worms that we use tend to be smaller. Would you like to see them? I happen to have them right here. Okay, this is when I make a big mess. Okay, so these are red wigglers. And you see they're very small. And they wiggle, no. But that's how they got their name. And the Latin name for those is Acinia fetida. And that's the kind of worm, they're really excellent at composting. Okay, so now I made a huge mess. I'll clean it up later. But anyway, so those types of worms are what you use for composting. They're not native here, so you can't really have them outside, although I have a worm bin outside and I heat it with a seedling mat that I have temperature controlled to 40. If it drops below 40, like now, it heats up. And the worms hang out at the top anyway, and so that and everybody survives the cold winters. So they can't handle below 40 or above 90. Those jumping worms also can't. The large, and they're like this big, imagine compared to the size, they'll die, but their cocoons can overwinter. Hopefully not minus 14 degrees, but we'll see.
And then last one I see in the chat right now is from Suzanne asking the difference between butchering my words today, inoculant compost and matured compost.
What was the first word?
The difference between inoculant compost and matured compost.
When I think of inoculants, I think of compost tea. And what I think that's what Alex was referring to, that he's going to use that compost to make a tea. Because it doesn't have this mature soil structure to it, it's not going to add the soil structure to, but it does have a lot of life. And so by taking that compost, and I have a wonderful example, hopefully I'll have time to show you, of when we took fungal compost from around some really healthy mountain laurels, and we made a compost tea of that. So we had a high fungal compost tea, and I sprayed it on my diseased mountain laurels, and guess what? That black spot and the rust went away. It was quite amazing. And they're doing much better now. So we're looking at using these inoculating teas to do a number of things. Everything from, sorry, I got a cough for a second. From being able to give you immediate plant available nutrients to being able to fight certain diseases and pests. So what we have often used the compost tea, I remember spraying it on a row of peppers. By the time I got to the end, the ones at the beginning were literally large, bright, greener, standing up. I'd seen an immediate difference. But I'm getting more and more into composting, and I do want to talk a little bit about regenerative ag principles that can keep adding to the microbial population and the maturity of your soil to make it as much life as possible. So let me stop sharing this screen, and now I'm going to share my PowerPoint again. Yeah. Okay, hopefully you can all see that. That's a big reach. Okay, so, here we are talking about compost tea. And just look at the difference. And what it is, because I don't even know if I told you this, it's aerobically brewing compost tea in water with air going through it. And then you add amendments. In this case, I usually add liquid kelp, fish hydrolysate, and some humic acid. The microbes eat those amendments, multiply by the billions within 24 hours, and then that tea is what you apply, foliar and around the roots of your plant. This is Dr. Ingham doing an extract in this case, where she took some compost, and you can do this very easily. Take your compost or worm castings, put them in a 400 micron mesh bag. That's the right size to allow the microbes to go through, but holds most of the material. So you would put that in there. The microbes are released into the water. They're not going to multiply as they would with a compost tea, but they'll certainly be in the water, and then you add that solution to your plants. So here's an example of compost tea, and this is what I used to do in the beginning stages, is just take a 5-gallon bucket, put an aquarium pump in there, and then just with a nylon stock or cheesecloth, the compost or the worm castings, the life will go out into the brew, and the things will multiply. Don't use these air stones, by the way, because they'll end up getting a biofilm on them, and that's not good. But that's a whole other story. So this is something that we just talked about. Adding the oxygen is adding with a pump of some sort, and the microbes consume the amendments and multiply. So, all right, so I'm going to show that short 1-minute video of what that looked like, the compost tea made from high that high fungal soil that was found around the roots of those healthy mountain laurels.
This would do really well.
And this is cooling down now. It's been.
No, that's not what we're looking at. Sorry. Okay, here we go. So this, let me start from the beginning. All right, here we go. So we're looking, this is some fungal hyphae here, just to show you. This is another type of fungi that we found in an untilled bed, garden bed. And this is what we found in that soil around the mountain. Look at the diversity of that fungi. I had never seen such beautiful, a little bit of a geek here, but I've never seen such beautiful fungi. And so I wanted to share that with you. And this looks like Bigfoot in here, but it's interesting shape, full of fungi as well. And so we took this, we made a tea, and we brewed it 24 hours. And then I'm going to show you in a minute what that compost tea looked like. So we're still looking at the actual soil here. And this is the compost tea. So 24 hours later, and I was really thrilled to see that the fungi had held its integrity in terms of hadn't broken into tiny bits for the most part, and could then go into the soil around the mountain laurels and introduce that fungi to form that symbiotic relationship with, in this case, the beautiful mountain laurel shrubs. All right, so I'll stop that. And I'm not sure if there are any more questions. We're getting short on time here, but I'm going to go back to my presentation. And I have to find it. Bear with me here. Okay, so are you seeing the regenerative ag principles, the cover crops, companion planting? Excellent. So we're wrapping up. We have a question from Jay. You want to unmute yourself?
Yes, I do. Hang on just 1 second.
Sure.
Okay, so I'm here and I'm relatively new to New York, and I grew up in Nepal where we have a 12-month growing season. However, here in New York, obviously, it's a much shorter season. So for the soil, is it fine? I mean, how do we help it during the winter time? Are these things perfectly adaptable to a 6-month growing season? Is there a way to make the soil, continue to improve with our crazy weather here?
The crazy weather is right, Jay. So my suggestion is, and we talked about in the beginning, try to have something growing. So for example, winter rye would be a good thing to have as a cover crop. Perennials, that's why I'm planting more and more perennial crops like asparagus and raspberries and strawberries and horseradish, all the things that will stay in the ground so that it will cover and those roots will continue to work. If you are in the fall, we're now moving away from pulling everything out, making your garden all tidy. I don't even take the seed heads off anymore because the birds love the seeds. I leave everything, and the roots that are in the ground that are annuals will slowly decompose and be food for those microbes. The microbes will go dormant in really cold weather, but the soil will be covered, and when it does warm up, those microbes will have ready food for as the plant's roots decompose. And the top, I'll lay that down, and that's a mulch that goes on top of the beds before I plant in the spring. So I hope that answers your question. Again, as much as you can have living as possible, always keep your soil covered. Never have bare soil, especially in the winter. Okay. All right. One of my favorite places in the world is Nepal. Okay. So anyway, regenerative ag methods. These are all things that really help with the life in the soil. So cover crops, we talked about a little bit. One of my favorites is to grow oats and rye and peas, and I'll harvest the pea shoots and eat them while the others are filling in. Again, covering the plant. I just mentioned that. Never let your soil stay bare, and covering with plant material is the best. again, the cover crops build soil health, nutrient retention, erosion control, and will stop the weeds because there's something else healthy growing there. I talked about peas and oats, and that the nitrogen fixing that goes on with those. The next thing to talk about, I'm rushing, but I just want to get these points across. Companion planting, plants that help one another. So for example, if you're growing tomatoes, using the shade of the tomato to grow the basil, and they go together and then they help one another. Marigolds is a wonderful companion for a lot of plants. So there's things that really do. I've got a book called, oh, there it is, Carrots Love Tomatoes, which goes into detail about that. And again, you might have heard of the Native American Three Sisters garden, where every plant helps the other. So the corn grows and the pole beans use the corn as a trellis. The squash around the bottom will protect the roots and cover the soil. So they all add and work together. Crop rotation is also a way to increase the viability of the soil. Changing what you grow in an area is always very helpful. And for example, if you're growing brassicas, they're going to take a lot of the nitrogen out of that soil. So following it with a high nitrogen legume like a pea or a bean will add that nitrogen back into the soil, especially if you use it as a cover crop and let it decompose in the soil. And then succession planting. So if you're, say, growing basil, plant basil every 2 weeks, or especially things like beans, because beans, your bean plant is all going to be mature at the same time. So by planting it in succession, you will be able to harvest, and you will also cover the soil and have different rates of growth at different times. The other thing is mixing up what you're growing. So start plants, you can do 3 types of plants in one bed. So you'll start with plants that can handle frost. As you're harvesting those, you're putting in the seedlings of the summer plant. As you're harvesting that, you might lay down a cover crop or a fall crop. So adding that, intercropping as well. So as I talked about, plants that don't compete, but actually will be companions with each other. And then, as we talked about, putting the same crop at timed intervals rather than all at once. There's a wonderful example at a school garden in Newport. Okay, I'm going to stop sharing. I have 3 minutes left.
Awesome. We have a couple questions. Let me go back in the chat. Suzanne asks, have you looked at soil which incorporates biochar? Are there noticeable differences or improvements?
Oh, good you asked that question. That's my test trial this year. Because the advantage of biochar, there are so many cell walls and areas for microbes to live. My question is, how am I going to see them if they are in the biochar? But I think what we're going to do is add biochar to one bed and not the other. Look at what the effects are above ground, and then look at it under the microscope to see if that housing for the in the biochar allows and increases the microbes. So stay tuned. That'll be this year's test.
Next one would be from Glenda saying, didn't Monique say she had applied the tea to leaves of laurel or also to soil?
I do both. So I will do a foliar spray. And again, when you're spraying, make sure it's not like a jet that's just shooting out, because those poor microbes they're not going to be happy. And it has to be wide enough that droplets can form so that it's not like a mist. So figuring that out so you can see the leaves sticking, and you'll see it actually does stick to the leaves. And then I usually then put it in a watering can and water around the roots of that plant.
Next one would be from Dawn asking, is there a way to rescue worm compost that may have gone anaerobic?
Good question, because I've done that. What do you do with anaerobic worm castings? What you can do is take some high carbon like wood chips or soil that's around your old growth forest. It'll tend to have a lot of oxygen in it, and mix that with your anaerobic or stinky pile. Don't use it right away. It'd be great if you could, get it under a microscope to take a look. But if the smell goes away and you start getting a better consistency, depends how far gone it is, you might be able to, and with the more fungal dominant soil, you'll have different microbes in there that will hopefully take over when the ones that like anaerobic conditions no longer have those conditions. But it is a process and takes some time. But you can do it.
Awesome. And then next one would be from Carrie asking, what about leaving old crops with regard to disease and such? When we were talking about like winter cover.
Right. Well, if you have a late blight on your tomatoes, you don't want to leave those. You don't want to have anything that is diseased. But if you have healthy plants, which, the healthier your soil is, and the more microbes you have, though, the healthier you'll tend to have your garden, you won't have to worry about that. So that you can, if you see disease, remove it, yes. But if everything is healthy, it can just healthily die and decompose right on the spot. Less disturbance is better. So I've gotten so neurotic now. Neurotic, maybe I don't know the best word. But anyway, diligent, shall we say diligent, that if I have a tomato plant and I'm toppled it over, I'm still not going to disturb where the roots are in the spring. I will plant around it and allow the integrity of the decomposing roots to continue. And I'm not disturbing the soil where there are roots.
Next one is Susan saying, thank you to the answer about the biochar. She asks, where will we see the results of your biochar study?
How about NOFA New York Winter Conference 2023?
Awesome. Megan M, this is more to me and the NOFA staff. Where's the survey for this session? Megan, if you, in the chat, if you go back to the 11:06 a.m. marker, Heather has dropped the survey link in there. So I think it's still in there. let me repost.
I got a thumbs up from Megan.
Okay. Yeah, let me repost that in here, make sure it goes to everyone. So we all have the survey link. And just to preface this, the survey link is to help us improve about what went right, what could be improved about the conference sessions. And we use all this data internally just to fine tool the conference. So next year's conference is even better. So if that's for y'all. Next question from Dave would be, would you say that the compost we examined was an example of high or low soil life?
I was missing the larger predators, but I can understand this time of year, it doesn't have, there isn't that nutrient cycling going on with living plants because it's compost on its own stored. So it had great soil structure, great aggregates, nice combination of fungal bacteria. So I think it's going to do great. And I'm sure once it gets on the soil, those smaller predators will show up and continue the work. So I'm happy with it for considering it's freezing and January.
Evan asks, would it be effective to cut back your annual crops and then cover with silage tarps before winter?
I would use silage tarps more for killing, for example, if you have a cover crop, say you did winter rye. In the spring, I would cut it really as down low to the ground as possible and cover it with the silage tarp to kill it from continuing growing. Otherwise, that's all you're going to be growing there is winter rye. It's a really prolific cover crop. But that silage tarp should kill it. And so it will, and then you'll have residue, and then you can plant into that.
Next one from Judy is, Beth best methods to create higher bacterial compost, less high carbon input?
Yeah. So bacterial does go along with, so if you're adding a lot of food scraps and things, you'll tend to have more bacterial. Steel cut oats, if you do want to get a fungal, say you want to make a tea and you don't really have the fungal in your compost, adding some steel cut oats to that compost, let it sit for 2 weeks, it'll form all the fungal and use that to make your tea. So there are cheats to make compost tea that is higher fungal. And as you mentioned, more bacterial. But generally, you don't have to worry. There's enough bacteria in everything. It's the fungal that's usually missing.
Yeah. And then the last one I'm seeing in the chat for right now is from Judy asking, they're saying late harvest make it way too late to sow cover crops. What is the best way to mitigate?
Late harvest?
Yeah, she's saying that
What to do?
Late season, make it to fit in a cover crop. What would be the best way to mitigate?
So if you don't have a cover crop, some people will put raw manure down in the winter, and that will slowly break down. As long as you have 120 days before harvest, you can do that. That's a good way to cover the soil. Some people will put compost down in the fall and have it slowly break down. Generally, I will take whatever is living there or I've harvested whatever it is, and just let it decompose, as I mentioned, so that I'm not disturbing the soil and it is covered. If it's not covered, just put down some type of mulch to cover the soil. Don't let it be bare if you can.
This is a really interesting one because I've been thinking about this personally. Sue asks, if you're just starting out on land, would you use silage tarps to start your fields versus tilling to prep?
If I were just getting a new piece of land, I would till and add whatever amendments are especially minerals, show up as being deficient in your soil test. Soil test first, till it once, add the amendments, including organic matter if it calls for that, and then that's it. Never till it again. It's the most effective way to really get in there and make that change, improvement.
Thank you. Awesome. I think that is everything for the comment thread questions. If I've missed any of the questions, feel free to unmute yourself, raise your hand, and we can answer any other questions. Or if you don't have any questions, feel free to fill out the survey on your own time and have a wonderful rest of this late morning we have here.
Thanks. I really enjoyed that. I was so nervous, but you guys were awesome. Thanks.
Yeah, that was a fantastic presentation.
Thanks.
Just a quick note. NOFA New York is working with Monique on a small grant project. So if you have any further questions or desire to be part of some of this, why feel free to contact us, and we'll put you in the right place with the right person to make it happen.
Right.
Monique has helped us out here on several occasions putting this forward, so appreciate it.
I appreciate all the work you're doing, Robert. Thank you. Okay.
Awesome. If there are not, if there are not any other further questions, I'm going to stop the recording here.